All right, so surface glycoproteins form ID markers for the cell. Again, this identifies a cell as belonging. This tells your immune system, for example, when to leave a cell alone or to attack a cell. These are made of, or they have what are called oligosaccharides on them, which are short chains of sugar. Your blood type is determined by these, as I mentioned previously. And again, an oligosaccharide would look like this. It's a short chain. These would actually be hexagons. And it would sort of look like that. And they have specific shapes. So you would need to match somebody in order for you to, for example, receive an organ transplant. So your immune system would attack cells that don't belong because they would be missing the proper ID markers. All right, these are some pictures. So on the first picture here, this is what transport proteins look like. When they want to indicate that one of them requires energy, they'll actually draw, this is like the big key, they'll draw ATP here, showing that energy is needed for this to work. Enzyme proteins, just speeding up a reaction. Here's an attachment one that's attached to the cytoskeleton. A glycoprotein, in this case, that's attached to another cell. This would be a junction, like a tight junction, for example, and this is what a receptor would look like. So again, really the proteins are these long folded up chains, but this is how they're usually represented in the cell membrane as simple like ovals and things like that. All right, so selective permeability. What can and cannot pass through the membrane? What can pass through the membrane, basically anything that's lipid soluble can pass through the membrane because phospholipids are mostly fatty acids. Those long fatty acids chain make up way more of a phospholipid than the little phosphate group does. So lipid-soluble things like vitamin E, for example, goes right through your cell membranes and directly into your cells because vitamin E happens to be a lipid-soluble vitamin. Small molecules like oxygen and carbon dioxide, because the membrane is fluid, those very, very small molecules can pass right through. They always list water, but technically water does not pass through the phospholipid bilayer. Water actually passes through special channels. And it sounds like I made the word up, but I did not. It's aquaporins, like water pores. They're literally protein channels that do nothing but allow water to pass through. Energy is not required for that. Water will naturally flow just like a tunnel. So water can go in both directions through these freely. For the most part, nothing else can make it through the phospholipid bilayer. When they say it's selectively permeable, what they really mean is that the membrane pretty much blocks almost everything. Most things have to get in and out of the membrane through the proteins. They're the most important part. So large polar molecules like sugar, for example, cannot pass through the phospholipid bilayer. And your cells need sugar or they starve to death. So the only way it gets in is through the proteins that carry it, not through the phospholipids. Additionally, ions, which again, your cell needs. Your cells, I mentioned in the past how muscle contraction relies on calcium ions. And nerve impulses rely on sodium and potassium ions. And so those things cannot, they're repelled by the lipid part of the membrane, but they can pass through proteins. But you have to have the proper proteins present to carry them across. This little diagram sort of shows you what can and can't pass. So really big things are not going to fit through the membrane. They're literally going to have to be eaten by the cell if they're going to be taken in. Something really small like this molecule is probably supposed to be small or lipid soluble. It can go through. And this third one is probably supposed to be like glucose. And it requires a protein to carry it through. So most things don't naturally flow through the membrane. It provides a barrier that controls what enters and leaves. Only things that are lipid soluble and very, very small. Everything else relies on proteins to determine if it's going to get in or out. All right, and to wrap things up, so we're going to be spending pretty much the rest of this chapter is on cell transport. We're only going to do 2 definitions today, and those 2 definitions are for diffusion and osmosis. So diffusion does not rely on a cell or energy or anything else. Diffusion is, and I'm sure you know this definition already, it's just the simple movement of molecules from higher concentrations to lower concentrations. This does not require energy because molecules are always moving. So the kinetic energy that the molecules already have within them is enough for diffusion to occur. If I sprayed perfume in the corner of the room, after a few minutes, other people across across the room start to smell it. That's diffusion. The perfume molecules sort of get knocked around, and eventually they end up spreading out. They're not purposely going towards anybody on the other side of the room. They just happen to get bumped around and end up over there. If I put food coloring in water, I see diffusion too. Again, the food coloring molecules get knocked around by the water molecules. Eventually, everything ends up spread out. Osmosis is just the diffusion of water. So technically, osmosis is the same as diffusion, but specifically referring to water. Water will also go from higher concentrations to lower concentrations without any extra energy being added. And that's basically what we're going to be doing in the lab. We're going to use dialysis tubing to make little bags. We're going to put certain things in the bag, certain things outside the bag, and we're going to see what can diffuse through the membrane and which way will things go. Anytime a process does not require energy, it's called passive transport. So, and again, passive transport happens because it's going specifically from higher to lower concentrations just by the random motion of molecules getting bounced and knocked around. They will go from being more concentrated and end up in areas where they were less concentrated until everything pretty much just reaches equilibrium. And you have similar amounts going in every direction, and that's what we call a dynamic equilibrium, because stuff doesn't stop moving, but in general, the amounts going in every direction are the same. So the concentrations don't actually change. The last thing I'm going to show you is just a picture of diffusion, just to clarify what it looks like, and then that's where we're going to stop. So if you still need this, I'll come back to this slide after I show you the picture. That's actually just an example of osmosis with some water traveling across. But here's what they mean by diffusion. So, and I think this, if I can stretch it, there we go. So if we look at the beginning, you've got some molecules of dye on the left side, and on the right side it's just pure water. And this is supposed to be a membrane. So stuff can pass in both directions through this membrane, but the dye is going to tend to go to the right because there isn't any dye on the left. So you're going to start to see the dye molecules, every time one of them hits a spot where it'll fit through a hole, they're going to start traveling towards the right side. Now you'll notice in the second picture that this dye molecule is actually going the other way, because the dye is not being sucked to the right. The dye is just randomly moving around. But the chances, since there's so much of it on the left, the chances that a piece hits from the left and goes through is going to be much higher than the chance that a piece on the right hits and goes through. And so that's what's going to happen, is you're going to see a net movement to the left, even though technically some of the molecules may end up moving the other direction. I think I'm mixing up my left and right, by the way, so just ignore my directions there. In the last one, we see what's called equilibrium. Basically, eventually, you have similar amounts on each side, so the chances of one of them going to the right and one of them going to the left are very similar, so you're not going to see a change in concentration. You're going to see similar amounts of dye on each side. Even though it hasn't actually stopped moving, it's still going in both directions, but at the same rate. So that is what I wanted to show you. It's just sort of an illustration of what's happening in diffusion. So we're going to stop with that.